Hey everyone, ever wished you could just hit copy on a strand of DNA? Well, that's basically what PCR lets us do, and that's what we're diving deep into today.
It's true. It's a game changer for so many fields.
We're using this great article from Assay Genie called PCR Types and Applications to explore all the ins and outs of this amazing technique.
It really is amazing. PCR is like having a DNA photocopier, right? It lets us make millions of copies from just a tiny sample.
It's mind-blowing when you think about it, like being able to study a gene from a single cell.
Exactly. And the process itself is fascinating. The article breaks it down into three main steps.
Denaturation, annealing, and extension. I always like those words.
They sound pretty cool, right? But they describe a very elegant process. Think of it like baking a cake.
Baking a cake. Okay, I'm listening.
So first, you gather your ingredients.
Makes sense.
In PCR, those ingredients are our DNA sample, something called primers.
Primers.
Yeah, and a special enzyme, DNA polymerase. It's the key to the whole thing.
Okay, so what are these primers? I'm not familiar with them.
So primers are like little flags that mark the specific DNA section we want to copy.
So they're like giving directions to the DNA polymerase, like start here.
Exactly. They're short DNA sequences that bind to the target region.
I see. So that's how we make sure we're copying the right part of the DNA.
Yep. Now imagine preheating your oven, separating that cake batter. That's like denaturation.
So we're using heat to separate the two strands of the DNA double helix.
That's it. It's like unzipping the DNA.
I love that analogy. So now we have two single strands of DNA ready to be copied. What's next?
Next comes annealing. Think of it like the primers attaching to their targets.
Oh, okay. So we lower the temperature a bit, and the primers bind to those separated DNA strands.
Exactly. They're like marking the starting line for our DNA polymerase.
Okay, so the primers are in place. The DNA is unzipped, ready for action. What happens in extension?
Now, this is where the magic really happens. The DNA polymerase comes in, grabs those nucleotides, and starts building new DNA strands.
It's like pouring the batter into the pan and baking, right?
Right. Perfect analogy. And with each cycle, the amount of DNA doubles. Denaturation, annealing, extension, repeat. It's really remarkable.
That's incredible. We're basically exponentially multiplying our DNA sample.
We are, but there can be challenges.
Oh, like what?
Well, sometimes you run into something called GC-rich sequences.
GC-rich sequences, what are those?
They're regions of DNA with a higher proportion of guanine and cytosine.
Ah, the G and C nucleotides. Right, I remember those.
Yeah. And the bonds between G and C are strong, really strong, which can make them harder to separate during denaturation.
So it's like trying to unzip a really stubborn zipper.
Exactly.
I could see how that would be a problem. So how do scientists get around that?
Well, they've developed some clever techniques, like increasing the denaturation temperature.
Makes sense. Just crank up the heat.
Exactly. Or they add special reagents, kind of like adding oil to a sticky zipper.
So even with tricky sequences, we've found ways to make PCR work efficiently. Pretty impressive.
It is. It's a powerful tool. But there's another cool variation we should talk about. Hot Start PCR.
Hot Start. Intriguing name. What's that all about?
It's all about accuracy, really. Imagine spilling some cake batter before you even put in the pan. Not ideal, right?
Definitely not. A recipe for disaster.
Well, in traditional PCR, the polymerase enzyme can sometimes start copying DNA too early at low temperatures.
Oh, so that's the spilled batter?
Exactly. It can lead to errors and unwanted products, but Hot Start PCR prevents that.
How so?
It keeps the polymerase inactive until the reaction reaches the right temperature. It's like having a safety switch on our batter dispenser.
Smart. So no more accidental copying and no more mutzy spills.
Exactly. We're constantly improving the precision of PCR. And speaking of precision, have you heard of high-fidelity polymerases?
I have, but I'd love to hear more about them. They sound pretty important.
They're essential for accuracy. Imagine you're copying a super important document. You want to make sure every word is correct.
Of course. Every detail matters.
Well, high-fidelity polymerases are like super careful proofreaders.
They catch every little typo.
Precisely. They have this amazing ability to double-check their work and fix any errors during DNA replication.
So it's like having a built-in spell checker for our DNA copy machine.
That's a great way to put it. They ensure we're getting those perfect copies, which is crucial for things like cloning or genetic engineering.
I see. So for applications where accuracy is absolutely paramount, these high-fidelity polymerases are a must-have.
Absolutely. They're truly remarkable enzymes. But, we've been talking about copying DNA this whole time. What about RNA?
That's right. RNA is just as important. So how do we apply PCR to RNA?
Well, there's a technique called RT-PCR. It's like translating a book before making copies.
Oh, intriguing. So we're
Yeah.
converting RNA into a language that PCR can understand.
Exactly. We use an enzyme called reverse transcriptase to turn that RNA into DNA. We call it cDNA.
cDNA. Got it.
Yeah.
And then we can use regular PCR to amplify it.
Yep. It opens up a whole new world of possibilities. We can study and work with RNA just as easily as DNA. It's a pretty big deal.
It's like having a universal translator for the molecular world. This is all so fascinating. We've covered so much ground already. The basics of PCR, those tricky GC-rich sequences, and even how to work with RNA.
It's just the tip of the iceberg.
I can't wait to delve deeper into the world of PCR. We'll be back soon with part 2 of our deep dive, so stay tuned.
Welcome back, everyone. We're ready to dive even deeper into the world of PCR. Last time, we explored the basics, and now it's time to look at some even more advanced techniques.
It's going to get even more interesting. We're talking about real-time monitoring, super precise quantification.
Right, we left off talking about RT-PCR and how it helps us work with RNA. But today, we're tackling qPCR and isothermal amplification.
Exactly. These techniques take PCR to a whole new level.
Okay, so qPCR, the Q stands for quantitative, right? So we're not just copying DNA, but actually measuring how much is there. You got it. qPCR is like having a molecular counter. We can actually watch the amplification process as it happens.
So instead of just knowing if our target DNA is present, we get to see exactly how much of it is there in real time.
Exactly. It's like watching your cake rise in the oven, but at a molecular level.
I love that analogy. Okay, so how does this molecular counter actually work?
Well, it uses fluorescent dyes or probes that light up as the DNA replicates. The more DNA copies, the stronger the signal.
So we can actually see the amount of DNA increasing with each cycle of PCR. That's incredible. What are some of the ways scientists use this real-time monitoring?
Oh, it has tons of applications. It's used in medical diagnostics, like measuring the viral load in patients with HIV or hepatitis C.
So it's not just about detecting the virus, but actually understanding how active it is in the body.
Precisely. And that's crucial for monitoring disease progression and treatment effectiveness.
That's amazing. It's like having a window into what's happening at the molecular level. And the article also mentioned RT-qPCR. Is that related?
Absolutely. RT-qPCR combines the power of reverse transcription with qPCR. Remember how we convert RNA to cDNA?
Right. We use that special enzyme, reverse transcriptase.
Exactly. Well, with RT-qPCR, we can measure the amount of RNA in a sample after converting it to cDNA.
It's like a two-step process, translate, then quantify. That's pretty ingenious. It opens up a whole new set of possibilities for studying RNA, right?
Absolutely. We can look at gene expression, analyze RNA sequences, you name it. It's incredibly versatile.
So qPCR and RT-qPCR are all about precision and real-time monitoring. But what about this isothermal amplification? The article mentioned it doesn't need those repeated heating and cooling cycles.
That's right. It's a real game changer, especially for settings with limited resources. No need for fancy thermocyclers.
Hold on. So we can amplify DNA without constantly changing the temperature.
We can. Isothermal techniques work at a constant temperature. Think of it like baking a cake, but it doesn't need an oven, just sits on the counter and bakes perfectly.
That's amazing. No more bulky and expensive equipment. Are there different types of isothermal amplification?
Oh, yeah. One popular example is LMMP.
LMB.
It stands for loop-mediated isothermal amplification. That's quite a mouthful.
I can see why they shortened it. So how does it work?
It uses a special enzyme and some clever primers that create loop structures in the DNA. This allows for continuous amplification at a single temperature, usually around 65 degrees Celsius.
So it's like a self-sustaining chain reaction, all happening at a comfortable temperature. I like it. The article mentioned LAMP being used for COVID-19 testing. That's pretty impressive.
It is. LMP's simplicity and efficiency made it really valuable during the pandemic, particularly in areas without access to those high-tech labs.
It's a great example of how scientists are constantly innovating and adapting these techniques to real-world challenges. It's really inspiring. But we can't forget about another groundbreaking technique, digital PCR. It's supposed to be incredibly precise.
Digital PCR takes quantification to a whole new level. Imagine splitting your cake batter into thousands of tiny ramekins and baking them separately. That's essentially what digital PCR does.
So we're running thousands of mini PCR experiments at the same time.
Exactly. And by analyzing the results of each tiny reaction, we can actually count the individual DNA molecules in the original sample.
Wait, we can count individual DNA molecules. That's mind-blowing. So it's not just about estimating the amount of DNA, but getting an exact count.
Precisely. And this level of precision opens up incredible possibilities for things like detecting rare genetic mutations or studying gene expression at super low
It's
levels.
like having a molecular magnifying glass. We can see things we could never see before.
You got it. It's a powerful tool for researchers.
This deep dive is just full of surprises. From simple copying to real-time monitoring and now counting individual molecules, PCR has come such a long way.
It really has. It's a testament to the ingenuity and creativity of scientists. And it's not over yet.
There's more.
Oh, yeah. We've got one last type of PCR to explore, one that takes us beyond amplification and into the realm of manipulating and editing DNA. But for that, you'll have to join us for the final part of our deep dive.
And we're back for the final part of our PCR deep dive. We've gone from the basics to some pretty mind-blowing techniques.
It's been quite a journey, hasn't it? And we're not done yet.
You're right. Last time we were talking about digital PCR and its incredible precision, but today we're going even further.
That's right. We're stepping into the world of PCR cloning. It's a powerful tool.
PCR cloning, it sounds like we're not just copying DNA anymore, but actually manipulating it.
That's exactly what it is. We're combining PCR with the ability to insert specific DNA sequences into vectors.
Okay, vectors. Remind me what those are again.
Think of them as molecular delivery trucks.
Delivery tracks. Okay, I'm intrigued.
They carry DNA into cells.
So PCR cloning is like loading up these tiny trucks with specific DNA cargo.
Precisely. We use PCR to amplify a specific sequence and then insert it into a vector.
And then what? Where do these molecular trucks go?
They go into host cells like bacteria or yeast.
Okay, I'm following. So the vector with our DNA cargo gets inside the host cell. Then what happens?
The host cell replicates, making copies of itself and, importantly, the vector with our inserted DNA.
So it's like we're creating a DNA factory, churning out tons of copies of our desired sequence.
You got it. It's a powerful technique for producing large quantities of specific DNA.
That's incredible. We're not just copying DNA anymore, but actually engineering it.
Yeah.
What are some of the applications of this? This has got to have huge implications.
Oh, absolutely. PCR cloning has revolutionized so many fields. Genetic engineering, biotechnology.
Give me some specifics. What kind of things can we do with this?
Well, for instance, we can create genetically modified organisms, produce proteins for therapies, study gene function in depth. The possibilities are vast.
So we're talking about engineering life at its most fundamental level. That's both exciting and a bit daunting.
You're right. With this power comes great responsibility, right?
Absolutely. Ethical considerations are paramount as we delve deeper into genetic engineering. But wow, this deep dive has been an incredible journey.
I agree. It's been amazing exploring the world of PCR. We started with those basic principles of DNA replication.
Right. And look where we ended up. From simple copying to engineering life itself. It's mind-blowing how PCR and its variations have impacted our understanding of life.
It truly is. PCR has become an essential tool in so many fields, from diagnosing diseases to developing new therapies. And who knows what the future holds?
That's what's so exciting. New applications are emerging all the time, pushing the boundaries of what's possible. We could be talking about personalized medicine, engineering crops for a changing climate, even space exploration.
It's a thrilling time to be involved in this field.
Absolutely. I hope this deep dive has sparked your curiosity and given you a new appreciation for the power of PCR. It's a revolutionary technology that's shaping our world in incredible ways.
It has been a pleasure exploring these ideas with you.
The pleasure was all mine. And to our listeners, keep exploring, stay curious. There's a whole universe of scientific wonders out there waiting to be discovered.